Hello, and welcome to Taconic Biosciences webinar entitled Enhancing Reproducibility in Humanized Immune System Mouse Models, Foundations and Tailored huSelect Strategies. My name is Philip Dubé, and I am the Director of Scientific Marketing at Taconic Biosciences. In my 9 years at Taconic, I have worked with dozens of academic, pharma, and biotech clients around the world to help them understand and integrate humanized immune system mice into their research projects. At the conclusion of today's webinar, we will have a live Q&A session. If you have questions during the webinar, please feel free to enter them into the Ask a Question box located at the bottom of your screen. If time allows, I will address those questions at the end of the webinar. Let's get started. Today I will talk a little bit about the foundations of humanized immune system mouse models, how they're constructed and how they're used, and provide a few examples of practical applications of these in drug development programs. Next, I will consider a few of the issues surrounding experimental design and reproducibility specific to humanized immune system mice, followed by a brief discussion of a new offering by Taconic Biosciences that we have entitled huSelect that helps to overcome some of these issues in your research. Humanized mouse models is somewhat of a broad term that can mean multiple different things. On one hand, mice can be humanized by inserting human genes into the mouse genome. These can be human transcripts, human mini-genes, partial knock-ins of human genes, or entire coding and non-coding sequences for various genomic regions. On the other hand, you can also humanize mice by engrafting them with a combination of human cells or human cells and human transgenes as well. So this could look like human tissue grafts, engraftments of various human immune cell populations, or stem cells. Today I will be focusing specifically on human immune cell engraftment, otherwise known as humanized immune system or HIS mice. Now, humanized mouse models are not perfect. It is never possible to fully humanize a mouse. But as the old adage goes, not all models are perfect, but some models are useful. And I'll try to point out throughout the course of the presentation some of the limitations and some of the reasons why you might choose to use a humanized immune system model in your research program. Humanized models form one end of a continuum of different types of mouse cancer models that help to bridge the translational challenges experienced by these models. On one hand, we have fully mouse systems. So these would be mice typically engrafted with mouse tumors or syngeneic tumor models. These have several different advantages as well as disadvantages. On the pro side, they tend to be quite inexpensive and amenable to high volume target screening. And they also, due to the availability of tools to modify the mouse genome, they are amenable to genetic modification. Now, on the downside, fully mouse systems suffer from lack of conservation. So human targets may differ significantly from mouse targets. And that may lead to the requirement of using surrogate drug candidates for your syngeneic program, which of course will add additional time and expense versus being able to directly use a clinical candidate. Furthermore, there are numerous differences between mouse and human immune systems. I'm calling out one in particular, which is altered effector functions due to species-specific differences in both Fc gamma receptors, as well as binding to various IgG isotypes. Finally, as I mentioned, they have a fully mouse immune system, and while this can be a benefit when probing the immune system, there are numerous differences between the mouse and human immune system. course, immunodeficient mice and engrafting them with human xenograft tumors, CDX or PDX, offers some real advantages, right? Here you can study directly human tumors because these are typically very immunodeficient animals. Anti-drug antibodies are typically not a concern. However, they do lack that human immune system component, and any immunodeficient mouse is still going to have residual murine innate immune activity, which can actually interfere with your test article, and I'll provide a few examples of that later in the presentation. Finally, in the immunodeficient background, there tends to be a very, very limited tumor microenvironment, which may be important for some studies. So humanized immune system models aim to bridge and resolve some of these issues. They're not perfect, but they do provide a model in which human immune cell targets are present. This allows you to directly test a clinical candidate within an in vivo system, and in many cases can provide a model with an intact human Fc effector function that will faithfully recapitulate the type of IgG Fc interaction that you would expect to see in a human patient. On the con side, these are very artificial models. If we're talking about hematopoietic stem cell based models, these are models in which the T cells that emerge within the mouse develop in that mouse context and are educated within the mouse thymus. Depending on the type of model you're using, there can be variable myeloid cell engraftment and limited immune lineages, which may be important depending on the mechanism of action of your drug target. Finally, one well-known limitation of humanized immune system models are incomplete B-cell maturation and incomplete humoral responses. Finally, in some of the more advanced models, these can suffer from graft versus host disease, which not only limits the usable time frame that you can study these models, but also introduces confounding effects due to that non-specific xenoreactive graft versus host disease reaction. At Taconic, all of our humanized immune system mice are based off of the super immunodeficient NOG background. Taconic has been collaborating with our Japanese partner institution, the Central Institute for Experimental Medicine, previously the Central Institute for Experimental Animals, for over 20 years in the development of the NOG portfolio and various methodologies to humanize and utilize these animals in studies. The NOG is a super immunodeficient mice. It is based off the NOD background that combines the SCID mutation, rendering these animals deficient in T and B cells, along with a truncation deletion in the IL-2 common receptor gamma chain, thereby leading these mice to be non-responsive to numerous cytokines, leading to multiple additional immunodeficiencies, including NK cell deficiencies and dysfunctional myeloid cell lineages. This level of immunodeficiency makes this a very good mouse model to engraft and accept human tissues. At Taconic, the two basic ways, and these are used across the world, are either to engraft these animals with peripheral blood mononuclear cells or with CD34 positive hematopoietic stem cells. At Taconic, we call these huPBMC NOGs versus huNOGs. And this is what I consider the base strain of two different types of humanized immune system mice. PBMC mice are used primarily for short-term studies. They allow for rapid reconstitution of human T cells in a mouse. They typically have non-functional B cells and typically are absent of most myeloid lineages. The reason why these are short-term studies is because these animals invariably will suffer from graft versus host disease, thus limiting the study duration. In contrast, HSC engrafted animals are amenable to longer-term studies. These can represent a more quote-unquote complete immune system with additional immune cell lineages. However, this requires a lot more time to produce, as these stem cells need to engraft and to differentiate into the multiple different human immune cell lineages. One challenge with most HSC engrafted animals is the lack of functional B cells and adaptive humoral immune responses. The protocol for engrafting via either of these types of cells is a little bit different. Again, PBMCs typically come from adult humans. These represent mature, fully differentiated immune cell types that are available in quite large numbers. These are engrafted typically via tail vein injection and can be used quite soon following engraftment, but are typically only used for maybe 1 to 2 months following that period at most. In contrast, hematopoietic stem cells are typically isolated from term pregnancies and are isolated from the umbilical cord vein. CD34 positive cells are isolated and purified. Animals undergo a myeloablation experiment to open up a niche for engraftment of these stem cells, and are engrafted, subsequently again injected. And in this case, it takes many weeks for human immune cells to engraft, differentiate, and expand into sufficient numbers to be able to run a study. Now, as I mentioned, one of the challenges with hematopoietic stem cell engrafted HIS mice is being able to support various immune cell lineages. Now, this is a chart showing the differentiation of various immune cell lineages and the various cytokines that are required to support lineage commitment down each of these arms. Since mice and humans differ, their mice will lack some of the critical cytokines to support differentiation of certain lineages, in particular the myeloid cell lineages. So in the absence of any additional cytokines provided, the default route for differentiation is typically down the common lymphoid progenitors, and typically you will see both T cells developing as well as B cells. As I mentioned, one limitation is that B cells typically fail to mature and will not typically undergo a significant amount of class switching, meaning very low levels of human IgG production. Following engraftment, we typically set a lower cutoff rate to define a successful engraftment as being 25%. Now, it's a bit arbitrary, but that is the number that we typically use. Although on request, we can fulfill lower levels of engraftment, and there may be some reasons why that might be beneficial for your studies. And we assess the level of engraftment or engraftment efficiency by doing flow analysis from peripheral blood. In the case of HUNOG, it is 12 weeks post-engraftment. And we quantify the total number of human CD45, the pan-hematopoietic marker, versus mouse CD45 to determine that percentage. We also look at numerous other markers in these models. I'm showing here just markers for B cells, T cells, and T cell subsets, showing that each of these populations are present. And the T cells include both CD4 as well as CD8 T cells. So a regular HSC engrafted NOG has some strengths and some limitations. There are numerous studies using these in both research and drug development that have shown it to be predictive and translational, specifically with regards to demonstrating function of human T cells. In particular, these have been used extensively to demonstrate efficacy for various checkpoint inhibitors. One nice thing about this type of model is that these tend not to exhibit any signs of graft versus host disease, and engraftment will tend to persist for the lifetime of the mouse. And I believe our oldest HuNog is well over 1 year post-engraftment, so they're very, very good for long-term studies. Now, despite this, some of the limitations, there tend to be very limited human myeloid cells present. There are either low or absent human NK cells. And as I mentioned, right, these T cells are maturing in a mouse thymus. So T cell selection is not going to be normal per se, and that does present some challenges. B cells, again, tend to be immature, and we tend to see absent or very limited class switching in B cell maturation. Now, there are some strategies to expand and extend the number of lineages that develop in such a system. I'm showing here some of our other transgenic versions of NOG that express various different cytokines that allow for that immune cell differentiation, survival, and function into multiple different lineages. So I'll just run through these quickly. The one that we have spent a lot of effort in characterizing and using is called NOG-EXL, which stands for extended lineage. This is a transgenic NOG that expresses human GM-CSF and human IL-3. This supports both lymphoid and myeloid cells. The myeloid cell population tends to be granulite skewed. We produce these off the shelf. We generally have a large live inventory of these pre-engrafted that are ready for study use. Compared to other models out there, these tend to have a longer lifespan, but they will tend to develop complications over time due to the presence of human phagocytes. Some of our other models include IL-6, IL-2, and IL-15 transgenics. Each of these are used for specific purposes. IL-6 in particular tends to support more monocyte populations, whereas IL-2 and IL-15 NOG have much more specialized uses and are typically not used for hematopoietic stem cell engraftment. So in the IL-3 and GM-CSF expressing NOG-EXL, this is typically what we see following engraftment. We'll see a nice healthy population of human immune cells present. This includes both B as well as T cells, but additionally here we start to see the appearance and expansion of human myeloid cells. This is some data from a publication a few years ago that was comparing NOG to NOG-EXL, as well as an additional model, the NSG-SGM3, looking at engraftment kinetics and immune cell composition following engraftment. NOG-EXL mice tend to have a higher level of engraftment due to the addition of those human cytokines, but this also leads to a skewing and a differentiation of additional cell populations. So you can see a summary of some of the different types of cells and how they might compare to peripheral blood of a human patient. But most importantly, and why many people choose this particular model, is for the presence of myeloid cells. And it is noted in particular that they express a very significant population of plasmacytoid dendritic cells, as shown in this graph at the bottom. As I mentioned, these have been used extensively to study various drug candidates. These are some data from a publication looking at combination therapy of PARP inhibitor as well as a checkpoint inhibitor. I'm showing here first just the PARP inhibitor alone. So again, this is looking at the growth of a breast cancer tumor, in this case MDA MB436, within the humanized NOG-EXL. And here you can see a nice tumor growth inhibition induced by niraparib that is associated with an increase in human cytotoxic T cells as well as T helper cells in this particular model. Again, demonstrating how these human cells are interacting with that tumor in response to therapy. Most importantly, this model can then also be used to study combination therapies. So this is an example comparing niraparib alone or in combination with pembrolizumab, again, in that same MDA MB436 breast cancer in humanized NOG-EXLs. So together, these data just give you a few illustrations of some of the power of using humanized immune system mice in research and drug discovery programs. The huNOG-EXL has multiple different advantages. I mentioned availability. We engraft these routinely, so you don't have to wait 10, 11, 12 weeks to begin a study. We can often fulfill those orders much more quickly. I will describe later some of our efforts around pre-validating donors to increase your confidence in using these animals and to reduce variability and improve reproducibility. Each of these animals comes by default with a 10-color flow cytometry panel that is collected 10 weeks post-engraftment. We do have options to get access to animals earlier, and one of the reasons why you might consider that is that there is an early rise on myeloid cell populations. So if you're specifically studying myeloid cells, you may want to look at an earlier time point. We will provide HLA data, full HLA data for the human donors that are used to engraft these animals. And you do have access to animals engrafted with different hematopoietic stem cell donors. And again, I'll describe a little bit more fully how and why you might choose that later on. So humanized immune system mice are complicated models. There are many different considerations that may contribute to research success or failure. So we have learned a lot of lessons over the years. And I mentioned this earlier. This is the fact that all immunodeficient mice, NOG included, tend to have residual murine innate immune cells that can and will interfere with research results. So I'm graphically illustrating this here. The NOG mouse, like other immunodeficient mice, tends to have various mouse innate immune cells and mouse phagocytes that, although somewhat dysfunctional, are still present, and in particular can interfere by binding with, in this case, monoclonals through the FC region based on the FC receptors that are present on those mouse cells. This can lead to a lot of issues, including depleting cells, binding and sequestering your therapeutic, or even inhibiting human tumor-associated macrophages that might be present. So to dive a little bit deeper into this, mice have 4 different isoforms of the Fc gamma receptor, and they're not completely the same as the human orthologs. So humans have 4 different types of Fc gamma receptors, and their binding affinity for human IgG, the binding affinity of the mouse Fc gamma receptor for human IgG can often differ. And what that means is that it will potentially introduce unwanted Fc effector function, as well as unintended binding of human monoclonals when delivered to a mouse. Now, at Taconic, what we have done in collaboration with CIEM is to address this by knocking out both the common Fc receptor gamma chain as well as the Fc receptor 2b. And in doing so, we can completely abrogate all Fc gamma as well as Fc epsilon receptor function within that mouse. So this leads to an even more immunodeficient mouse that is unable to bind any monoclonals via Fc gamma receptor reactions. So why might this be important? So I want to show you just a few examples. Again, this is data from CIEM. On the left is a heterozygous animal expressing Fc gamma receptors, whereas the right is, in the graph, is a knockout for the Fc gamma receptors. And this is an example of getting rid of a false positive. On the left, in the presence of murine Fc gamma receptors, trastuzumab is able to induce a tumor growth inhibition, and that effect is completely lost in the absence of the Fc gamma receptors. Again, here suggesting that it is an interaction with the mouse innate immune system as opposed to the human immune system that is inducing this effect. So in order to be able to get rid of that false positive, to increase your confidence that what you're seeing is a human effect, we often will recommend using a model like the FC Resolve, which can also be humanized in a very similar way that I have described for the other models. Conversely, murine Fc gamma receptors can also introduce false negatives. On the top row here are 4 different types of tumors in humanized FC Resolve NOG, and the bottom are all just regular NOG that express murine Fc gamma receptors. You'll see here that the bottom showed only 1 tumor responding to nivolumab. In contrast, 3 out of the 4 tumors all responded to nivolumab with tumor growth inhibition in the absence of those murine Fc gamma receptors. Here again, giving you not only more confidence that what you're seeing is real, but actually enabling you to see that therapeutic response, because those Fc gamma receptors really can't interfere. Some of our internal data has actually demonstrated that this effect can actually be donor dependent. And depending on the HSC donor, you may see a different contribution of the murine versus the human response. So these are a panel of 3 different HSC donors engrafted with HCC827 CDX tumor cell line. And in the first case, donor A, we have essentially a non-responder or a very mild response, regardless of the genetic background of the mouse. In donor B, there is a response, actually a negative response. We see increased tumor growth with pembrolizumab in the HUNOG, and that negative result is resolved, getting rid of Fc gamma receptors. In this final case, donor C, we see a therapeutic response of pembrolizumab that isn't shown in the presence of Fc gamma receptors. And to me, this really brings up the really important issue of donor-dependent variability and how that impacts experimental results. So we have seen a lot of variability when it comes from one donor to another. So this is just another example of some of our internal data showing the level of engraftment of 3 different donors at 10 weeks and 23 weeks post-engraftment in HUNOG EXL mice. Although the levels at 10 weeks tend to overlap, you can see them diverge as these animals age and over time. Moreover, if you look at the exact immune cell profiles, so the human immune cells that are present, there are numerous different cell types that are quite different between donors. Now, of course, if you are running a study, this can and will introduce a significant amount of variation. And so, how do you deal with that? So one question was, can we engraft animals with aliquots of the same donor to eliminate that genetic variability between donors and see the same type of engraftment in the same cells present, or at least the same performance of the engraftment? These are data that Taconic presented in 2024 at AACR. This was a large tumor study with collaborators, and I'm only really going to show you some snippets of this poster. If you're interested, it is available on our website. In this particular study, we used 5 different donors. We engrafted one cohort 12 weeks before we engrafted the other. So essentially, we've repeated the exact same experiment twice using the same 5 donors, one aliquot of hematopoietic stem cells for the study for cohort A, and another aliquot for cohort B. The graphs in the lower right are demonstrating body condition scores for these engrafted mice following tumor engraftment, in which the B.Sc of 3 is a healthy mouse, and a B.Sc of 2 indicates declining health over time. And by just comparing the colors, it's apparent that there are similarities between each donor, but there are differences across the different donors. But importantly, those trends in how these animals actually performed remain somewhat consistent within that same donor over time. This led us to believe that we could use this type of approach to predict which animals would show a specific response and be able to go back to return to those donors and use them for subsequent studies, knowing what those characteristics would look like. So this is an example of one way that we have applied this at Taconic. We use this to screen donors to understand how well they will engraft in non-EXL mice. And then we will subsequently choose to use those for engraftments when possible. So this type of pre-validated hematopoietic stem cell donor is a really important technique to be able to produce a reproducible model with expected characteristics. So how do you deal with this in your studies? How do you deal with human hematopoietic stem cell donor variation? There are a number of different general approaches I'll summarize here. Some groups will want to use a single donor. Taconic, we do have the option to return back to that donor. We have demonstrated that we can produce the same engraftment characteristics on subsequent engraftments by going back to that same source. So one of the nice things with a single donor, this reduces a lot of variability. Now, of course, this does require that you're starting with a sufficient number of stem cells, right? They're a limited resource to support all engraftments for your needed studies. Now, although this has low variability, right, there is a risk that what you're seeing is inherent only to that one specific donor, right? And this might potentially question the external validity risk of your studies. Is it reproducible across multiple donors? An approach that some groups like to use It's certainly not one that I typically recommend, is by pooling donors. So if you take hematopoietic stem cells from multiple different donors, pool them into a heterogeneous mix and engraft that mix. Of course, this is one way of normalizing variation across all engrafted animals. By using a pooled sample, you can engraft many different types of individual mice from that one pooled sample. And although this may theoretically reduce and normalize that variability, I would point out that this introduces quite an unknown variable of having a heterogeneous mix of different human immune genetic background present within a single animal. And I think that raises perhaps a lot of potential questions about the validity of this approach. Finally, and this is typically my overall recommendation, is to use a donor-blocked randomization design, in which case for each donor, you would typically choose anywhere from 3 to 5, perhaps more, randomize each of those mice across each study arm. Now, of course, this will require not only more donors, but more animals overall, typically, to achieve statistical power. But one advantage here is that this can enable you to detect a donor-specific impact using a mixed effects statistical model, which is quite handy in understanding variation amongst the therapeutic responses from different patients. When selecting a HSC donor for your particular experiment, there's a number of different considerations. Some of these are practical, some of them are related to your specific hypothesis. First is very practical: cell yield and viability. These cells are harvested from umbilical cord vein. The yield can vary widely, as can the variability, and that will impact why you might choose that or choose to select another HSC donor. Of course, these are human samples. Each is going to have a complete repertoire of human HLA alleles, and in many cases, complete HLA information is available for these donors. So when thinking about HLA, you want to think about is a specific donor HLA required for your drug mechanism of action? Is matching to a tumor required? Now, I would point out, because the T cells that emerge in these mice develop within a mouse thymus typically, the HLA genotype of the donor does not necessarily indicate a HLA-restricted T cell response. But another reason why you might choose a specific HLA is to differentiate between the immune cells present and your tumor. For example, if you're working with a liquid tumor such as AML, multiple myeloma, etc., you might choose to specifically mismatch at one or more HLA alleles so that you can differentiate between that immune cell and the cancer cell. There may be reasons why you might choose a specific sex of a donor. So if you're working with a specific type of tumor, prostate, breast, etc., it may be advantageous to remove that as a variable, or to ensure that you are not biasing your data by selecting only one sex and not the other. And finally, at the end of the day, what matters is how these function. And one approach is to empirically determine how these cells perform in that mouse model system. So what does the engraftment look like? What do the cells that develop look like? And do they function? And I recommend, to have some type of predetermined inclusion or exclusion criteria for your study based on rate, presence or function of specific cell populations, as well as animal survival, welfare, and timelines. Now, at Taconic, we have long recognized a lot of these challenges, and we have worked to package them together into a suite of services that we call huSelect. These are essentially custom humanization services where we work with clients to ensure that we are providing the most appropriate animals for their studies, leveraging our technologies, our experience, as well as our very large inventory of hematopoietic stem cells. Now, hematopoietic stem cell donors, CD34 positive cells from these donors, these are finite resources, right? This only happens once in the lifetime of an individual. All donors have to go through stringent eligibility requirements, informed IRB consent, etc. And these are in demand across the world for many different research purposes. Storing these is a significant investment. As well as, with any primary human cell, there are always regulatory and ethical concerns and variability. At Taconic, we have assembled a repository of hematopoietic stem cells. Currently, our repository includes over 400 unique hematopoietic stem cell donors. I'm showing you here a snapshot of our inventory, including the typical detail that we have on HLA typing for each of these donors. Now, given that we have such a large repository, in general, we can usually match at least one allele for the major HLA loci, depending on how stringent your requirements are. Now, in huSelect, we give you, as the researcher, the ability to choose the HLA. Do you want a specific match? How stringent do you want that match? Is there a specific mismatch that you need for your particular study needs? We allow you to test out donors, either from our pre-validated stock, or you can test your own and return to them with subsequent engraftments. This allows you to be able to reserve donors within our inventory and come back to them to generate additional mice for subsequent studies. Additionally, we have built out significant capabilities in performing immunophenotyping via FACS analysis of these animals. So we can look into questions of, is a specific cell population present? Because of course, if you're studying a target on a specific immune cell, right, you want to know if it's there before choosing that particular model. Excuse me. So this is an example here of some of the pre-characterization data that we have generated for many of the different stem cell sources that are within our repository. This is looking here at survival post-engraftment or weeks post-engraftment, so we can have a good understanding of how long, in this case on a HuNog EXL, you might expect that animal to survive with subsequent engraftment. Chimerism rate, or the level of human immune cells that develop, as well as specific human cell populations, B cells, T cells, NK cells, and more. And you can see an example of some of these markers that we use here. Importantly, we also include information on the various myeloid cell lineages that are present. With our return to donor capability, again, as I mentioned very briefly, right, you can call us up and order a humanized mice to be shipped tomorrow. But if you're interested in using those to screen them for the presence of cells or a specific function, you can have us reserve additional animals from that donor, or if we have any cryopreserved inventory from that HSC donor, to enable you to go back to those animals and have better confidence that what you're seeing is going to be suitable for your studies. We have developed a number of different flow panels that are specifically designed to study humanized immune system mice. Our basic 10-color human flow panel assesses all major T, B, NK, and myeloid cell populations. We have various in-depth panels that include all human and B cell subsets. We have human myeloid cell specific panels, as well as murine immune cell specific panels. Again, because those murine cells can be present. So again, when trying to understand whether a specific donor or even just that type of HIS model in general is going to be suitable for your study, understanding what cells are present there is really critical. And the ability to go back to that donor is going to help you to give you better confidence that your target's going to be present. And, you know, if we happen to have enough cells, right, enable you to go back to it multiple times to repeat that experiment or perform additional experiments with the confidence that you're working with the same human immune cells present. Now, everything I've shown you today is from Taconic's products that based on our HuNogs or HuNog EXLs. But really, these types of custom humanizations are suitable for any type of immunodeficient animal. So if you have a specialized custom animal that has been developed for a specific purpose, right? Certainly talk to us, see if we can help you humanize it, leverage the capabilities that we have within our Humanization Center of Excellence to be able to provide you with, you know, a quality reproducible product that will be able to support your studies. So what that looks like is everything from breeding and mouse genotyping, determining control strains, understanding what type of myeloablation is necessary for your specific model, how many stem cells are required to get sufficient engraftment, and then finally doing an analysis of the time course of that engraftment, as well as the types of cells that are present. At Taconic, we take a consultative approach. Our team of scientific solutions consultants are available to provide complementary consultations on our humanized mouse portfolio, as well as everything that else that Taconic provides. So I would invite you to connect directly with a member of our scientific solutions team. You can visit us at taconic.com and contact us via that mechanism. And, you know, be more than happy to help walk you through some of the challenges and pitfalls that exist with humanized immune system mice. And with that, we will now begin the live Q&A portion of the webinar. So if you have a question, please go ahead and submit it in the Ask a Question box. And let's see, we have a few minutes for questions. Let me open up the questions and see what we have. Okay. Well, it doesn't look like we have any questions coming in yet. So with that, I thank you for your time today. I appreciate you attending our webinar. Thank you very much.